You're on mute.
Okay. Yes. Thank you. Thank you so much. Hi, everybody. Thank you for having me here. I'm really excited to talk to you about this topic that I'm obviously so interested and excited about. Let me just see if I can do 1 second. Took me a little while to figure out how to do presenter view on Zoom. So can you see my slides? Okay, great. So thanks for having me. I think we'll just go ahead and get started and then have plenty of time for questions and answers. Here's the agenda of what we're going to be covering today. So first of all, why even think about studying the gut in Parkinson's disease? We'll talk about what the function of the microbiome is. We'll talk about ways that we currently think that the gut and the brain communicate with each other, and then what we know about the microbiome in Parkinson's disease to date. And then a portion of this will be dedicated to just some recommendations about diet, dietary practices, and supplements for people with Parkinson's disease. And then we'll finish talking about this microbiome study that we're conducting here at Stanford, which for your group will be, part 2 of this on, like Monique was saying, on September 9th. So why study the gut in Parkinson's disease? I don't think I need to tell this audience that GI symptoms are really common in Parkinson's disease, and they start very early. So anywhere from like 60 to 80% of people with Parkinson's disease experience GI symptoms years and even a decade before their motor symptoms start, suggesting that the gut is involved very early on in the disease process, and it may actually be where the disease pathology for a lot of individuals starts. And so furthermore, that kind of gives us the clue as to why we want to be interested in the gut in Parkinson's disease. And then studies have shown that alpha-synuclein can be seen in the nervous system of the gut, which is called the enteric nervous system, really early in the stages of disease. And just to remind ourselves, alpha-synuclein is the protein that accumulates in Parkinson's disease. And so we get aggregates of this protein that are insoluble, and they become toxic to neurons and lead to neurodegeneration. So again, those aggregates of the abnormal protein alpha-synuclein can be seen in the enteric nervous system, the neurons that are in the gut, long before motor symptoms start. In animal studies, when animals have alpha-synuclein injected into the intestinal wall, it can travel to the brain via the vagus nerve. And then there was a Danish study from 1977 to 1995 that showed that when they did what's called a vagotomy, where they cut the vagus nerve, those people that had that vagotomy had substantially decreased risk of developing Parkinson's disease, suggesting that the vagus nerve may be critical for the pathogenesis or the development of Parkinson's disease. So what is the microbiome? The microbiome is a collection of microorganisms, and it lives both in us and on us. It consists of bacteria, viruses, fungi, and archaea. It's thought now that bacterial cells outnumber human cells by 10 to 1, and that we have 1,000 times more bacterial genes than we do our own human genes. We learned a lot about the microbiome and the function of the microbiome through the Human Microbiome Project. This was a National Institutes of Health research initiative, and it ran from 2007 to 2013. It cost our government $115 million, and it included 242 healthy adults, and they took samples from 5 different body sites. And really, again, the goals were to just kind of understand who's there in terms of bugs, what are they doing, and to start to understand the role that these bugs have in human health as well as disease. So from this study, as well as, of course, several studies since then, we've learned a little bit more about what the function of the microbiome is and how it impacts our human host physiology. Now we know that the microbes in our gut produce certain vitamins that we don't have the ability to make on our own. They help us to break down our food and to extract critical nutrients. The microbiome is very important in kind of training our immune system. It teaches our immune system how to recognize, bad guys and dangerous invaders. there's a lot of associations between alterations in microbiome and the development of autoimmune disease. The microbiome also helps to produce anti-inflammatory compounds, and oftentimes, then we'll talk about this more, in dysbiosis, or kind of a shift towards more inflammatory microbes, then of course there's an increase in inflammatory compounds that are made by the microbiome. The microbiome can promote healthy gut motility, and it also engages in something called bug-on-bug warfare, where the good guys, the beneficial bugs in our gut, help to fight off disease-causing microbes. And the picture here on the right represents one of my favorite facts from the animal kingdom. So this is that when baby elephants are born, they don't have the necessary microorganisms in their gut to digest plant materials. And so the only way for them to colonize their gut with the bacteria that they need in order to digest plants is to eat their mothers or other adult elephants in their herd feces. And so this is something that we see them doing when they're shifting from a milk diet to plant-based diet. Okay, so that's the microbiome and the gut. How do we think that this, interacts with the brain, and what are some proposed mechanisms by which the gut and the brain communicate? We talked about the vagus nerve, and there's actually a direct line of communication, meaning that the vagus nerve runs directly from the enteric nervous system in the gut into the brainstem. So that is quite literally a pathway or a highway from the enteric nervous system into the brain. Other ways that we think that the gut and the brain communicate is mostly through the molecules that the bugs in our gut make, and what impact those molecules can have on our either, systemic circulation or the brain. So some examples of this, the microbes in our gut make neurotransmitters. You've probably heard most of our serotonin is made by our gut microbes. Short chain fatty acids. These are molecules that are produced by gut bacteria when they ferment carbohydrates. Short chain fatty acids are, generally thought of as good things. They are fuel for the colonocytes, which are the cells that line the colon. And they've also been shown to impact the activation of the microglial cells, which are resident immune cells in the brain. So in short, the microbes make these metabolites. Two examples of this are short chain fatty acids and neurotransmitters. And those metabolites that they make have a direct impact on, other downstream effects, the way that neurotransmitters do, or they can alter the activation of immune cells. Other ways that we think that the microbiome has an impact or a kind of a way to communicate with the brain is through creating inflammation. And we'll talk about this because this is kind of a main interest in the study that we're conducting at Stanford. But inflammation in the gut can increase gut permeability, which can allow for microbes themselves, as well as those metabolites, be them good metabolites or bad inflammatory metabolites, to enter the bloodstream, where they can again lead to a cascade of events, including coming into contact with the brain and the blood-brain barrier. Monique, questions about that slide there while we're on it?
I don't think so. I think you did a great job.
Okay.
Could you just a quick overview again on what the vagus nerve is?
Yeah, so the vagus nerve is it's a cranial nerve. We have 12 cranial nerves. The vagus nerve is one of them. It's a parasympathetic nerve. It has many, many, many functions. My gosh, the vagus nerve is can be manipulated. We use vagus nerve stimulators in epilepsy. We don't really know how they work for epilepsy and why they impact seizures, but they do. You've probably heard in pop media that there are various things that you can do to kind of activate the vagus nerve, which can then activate the parasympathetic nervous system. But it's a cranial nerve coming out of the brainstem with direct communication into the gut and the enteric nervous system. And it has a big role in the parasympathetic nervous system, which is that rest or digest part of the autonomic nervous system.
Great, thank you.
Yeah. Okay, that was a heavy slide. Okay, so then in terms of microbiome disease, there's an ever-growing number of studies that have demonstrated that changes in the composition of the microbiome correlates with numerous disease states. Honestly, you name the disease, and there's some study that has shown that changes in the microbiome is associated with that disease or correlated with that disease. And this includes a variety of neurologic diseases, includes Parkinson's disease, everything from diabetes, metabolic disease, cancer, etc. So then it's also been shown that changes in the microbiome can be linked to various aspects of those diseases. So it could be associated with one's risk for developing the disease. It could be associated for how active the disease is and how it progresses over time, as well as the disease phenotype. So in Parkinson's disease, what we mean by disease phenotype is your disease motor predominant, tremor predominant? Do you have a lot of GI symptoms or not? So what do we know about the microbiome in Parkinson's disease specifically? It's pretty well established at this point that the composition of the microbiome in people with Parkinson's disease is different than people without. So if you just take a bunch of people with Parkinson's disease and a bunch of people without, and you look at who is in their gut, we know now that it's quite different. And the question that has started to be investigated in the more recent years is, what are those different bugs doing? And how is that, what functions are different as a result of the different composition? So these are just a couple of examples of what has been shown in terms of the different functions of those microbial species. So we talked about short chain fatty acids. Again, these are metabolites that are made by the microbes when they ferment carbohydrates. They're good, so we like short chain fatty acids. And generally speaking, people with Parkinson's disease have a decrease in the amount of bugs that produce short chain fatty acids, so therefore less short chain fatty acids. This generally is thought to be a decrease in anti-inflammatory things. And I mentioned that short chain fatty acids have been shown to alter the activation of microglial cells, which is a very important type of immune cell in the brain. And so that can shift the microglial cells to actually a more pro-inflammatory phenotype. It's also been shown that this change in the composition of microbiome in Parkinson's patients causes an increased production of inflammatory metabolites. So one of the products or the results of this change that we see in people with Parkinson's disease is they have more inflammation inside the gut. And then my favorite really interesting thing is that most of us, I think probably close to 100% of us have E. coli in our guts, but there's many different strains of E. coli. And there's a specific type of strain that produces this protein called curly protein. And it was shown that this curly protein that's made by the E. coli can cause alpha-synuclein to form these aggregates and therefore form these insoluble tangles that become toxic to the neurons. So that's a pretty compelling piece of evidence for how the metabolites that are made by certain bugs can have direct impact potentially on Parkinson's pathophysiology. So then the question becomes, okay, so we know that I might have, an altered gut microbiome. What can I do about it? This is just to remind us that there are many, many, many things that contribute to one's microbiome. So we can't be too naive in thinking that if we change any one or two of these variables, then that's going to, drastically change or permanently change our microbiome. And it also reminds us that when we compare individuals with Parkinson's and individuals without Parkinson's disease, there may be, of course, other factors that contribute to those differences. But that said, diet is one piece of this. So I thought, let's talk about diet. And this is always a popular topic with many questions, as it is very controversial as well. So with diet, we'll just talk about some general nutrition principles, and I'll talk about some dietary practices for individuals with gastroparesis, which is a majority of people with Parkinson's disease. With the caveat being that diet and nutrition are very poorly understood by science. And this is part of the reason why diet is so inflammatory of a topic to talk about, and so controversial. And probably for every argument of one way, you can find a counter argument to the other way. There's very poor quality of nutrition research. And so there's very little that we know for certain based off scientific evidence. And this is because epidemiologic studies don't derive causation. They cannot tell us that something causes X causes Y. They can show correlation, but not causation. And the majority of dietary studies are epidemiologic. And there are some clinical trials with dietary, looking at dietary practices, but the majority of them are pretty flawed by design. And so again, just kind of take everything with a grain of salt to some extent. Okay, so nutrition principle number 1 is just thinking about eating anti-inflammatory foods. So on the left is this standard American SAD diet with the sad face that we're all familiar with. And then on the right is actually Dr. Andrew Weil's anti-inflammatory food pyramid. So you may be familiar with him. He's kind of thought of as the grandfather of integrative medicine. He started the Integrative Medicine Center at the University of Arizona. And this is his anti-inflammatory food pyramid. And generally what you'll notice is that fruits, vegetables, non-processed grains, beans, legumes, and healthy fats make up the base of this pyramid. If you want to look into this more, you can just Google Andrew Weil, it's W-E-I-L, anti-inflammatory food pyramid, but we'll go into kind of some of a little more details on that. So in picking fruits, you want to look for lower glycemic load. So this is generally berries and then a bunch of the other fruits that I'll just let you look at here. In picking vegetables, it's what your mom or your grandma always told you. So you want a wide variety of colors on your plate. And this generally just helps you get diversity. Diversity in general in diet, but specifically in vegetables, because of the different types of fibers that they have, creates a more diverse and robust microbiome. And then here I say to avoid raw vegetables in individuals with gastroparesis or GI difficulties, just because raw vegetables can be pretty aggravating to the GI tract. They're harder to digest. They can produce a lot more gas and bloating. So if you're already having a lot of those symptoms from gastroparesis, then raw vegetables are best to avoid. In terms of proteins, understanding, some people here, of course, are going to be vegetarian or vegan. I personally recommend, a variety of proteins. So meat, red meat, chicken, salmon, dairy proteins. A couple things to think about. When eating red meats, you want to look as much as you can for grass-fed and grass-finished. It is more expensive. But if you're limiting your red meat, then really kind of, splurging on the more, the healthier version of red meats. Most of the studies that you see in the media that are showing, association with atherosclerotic disease or various inflammatory disorders linked to red meat are not grass-fed, not grass-finished, which means that they have a higher index of omega-6s compared to omega-3s, which tends to be pro-inflammatory. To my knowledge, there are no studies of how red meats impact any of that stuff when it's good quality grass-fed and grass-finished. Salmon, you want to try to get wild salmon. Sockeye is a really good thing to try to get. Sockeye cannot be farmed, so it's always going to be wild, and it has an especially good ratio of the omegas that you want. And if wild salmon isn't something that's readily available where you live, then you can check out Vital Choice. It's just vitalchoice.com. They're a really great seafood dispensary online. And then for fats, generally you want to try to get more unsaturated than saturated fats. Omega-3s and omega-6s are polyunsaturated fats, and you want more omega-3s than omega-6s. So that's going to be extra virgin olive oil, nuts, avocados, seeds. Omega-6s are more the vegetable oils. So canola oil, what else? Kind of the other sunflower oil is another example. So just to remind ourselves, omega-3 and 6 are essential fatty acids, meaning that our body cannot make them. So we have to get them from our diet. And I actually generally recommend that everybody supplement with omega oils. And this is because you can get enough from your diet, but for most people, it requires eating fish or other foods high in omega-3s like 3 to 4 times a week. It's just a lot. And I think realistically, most of us have trouble getting all that we need through diet. And there's pretty good data to show that higher plasma levels are associated with less brain shrinkage and a decreased risk of dementia. We just know that these are very healthy for brain health, very good for brain health. And so, yeah, that's why I recommend supplementing. There are tests out there. This is probably tangent, but there are tests out there now that you can get over the counter that it's just a finger prick of your blood, and it can test your ratio of omega-3 to omega-6 and tell you how deficient you are in omegas. I just learned about this and actually have just started to use it in my clinical practice. Okay, so nutrition principle number 1 was kind of the anti-inflammatory food pyramid. Principle number 2 is to eat organic when you can. It's expensive, so I know we can't do this all the time. But when possible, try to eat organic, and at least try to eat organic for the dirty dozen. So these are the things in the red here on the left. This is from the Environmental Working Group, or EWG's website. They're a great resource if you're not familiar with them. But basically what this says is that these things that you see in the red, the 12 pieces of produce here, are the produce items that tend to have the highest pesticide residue on them. And it's quite critical to eat those organic when you can. The clean 15, on the other hand, in the blue, are things that don't accumulate pesticides. And I think it's usually because of the skin, the thickness of the skin. I don't know why asparagus is on there, but I'm happy that it is, because conventional asparagus is far cheaper than organic asparagus. But generally speaking, these clean 15, you should feel okay not eating the organic versions of these, but really when you can, try to eat organic of the dirty dozen.
And doctor, when you are buying either organic or not organic,
Yeah.
it's important to wash your food before you cook it.
Correct. Yes. Yes, definitely recommend that. And you'll see, I'm sure people have seen, like, there's various ways. So there's produce washes that you can get in Whole Foods or wherever else. And then a lot of people use just white vinegar to soak certain produce things. If you Google online how to clean your produce, that's another option too. That I can't say that I do that with everything, but that's another option if you wanted to be really judicious about cleaning things.
Thanks.
Yeah. Okay, third principle is thinking about incorporating fermented foods in your diet. So Christopher Gardner is a professor and the director of nutrition studies at the Stanford Prevention Research Center. And he says that microbiota targeted diets, which in this case includes fermented foods, can change immune status, providing a promising avenue for decreasing inflammation. And this is based off a study that was done at Stanford, where they had individuals incorporate, I forget how many, I think it was 2 servings of fermented foods per day. And they measured the level of inflammatory markers in the blood and showed that individuals who ate fermented foods daily actually had substantially decreased inflammatory markers in the blood. And so fermented foods can include yogurt, kefir. I've never actually seen fermented cottage cheese, but supposedly it's out there, kimchi and other fermented vegetables, kombucha. And so these things can increase the diversity of our gut microbiome, and they're kind of like food for the good bugs in our microbiome. And again, we've now seen that you can actually have a decrease in inflammatory markers just from eating fermented foods. I had a question earlier about kind of, can you eat too much fermented foods? And we'll talk about this as it comes to probiotics as well, but some people will notice an increase in bloating, and just GI discomfort from eating fermented foods. I would say 1, start slow. So if you are not, used to eating fermented foods or not used to taking probiotics, then just introduce 1 serving a day for a while and increase that as tolerated very slowly. And if you're eating like 1 to 2 servings a day and you start to eat more and you notice that you have increase in GI symptoms, then just back off. So let your kind of symptoms be the guide. Okay, so that covers dietary principles. We'll move on to practices recommended for people with gastroparesis, which is, again, most individuals with Parkinson's disease. You want to eat smaller and more frequent meals. Remember, this is hard for me and probably a lot of us, but digestion begins with chewing. So chew your food very well. Fat can delay emptying of the stomach. For all the reasons we just mentioned, you don't want to avoid fat, but try to kind of spread it out across meals instead of having like, one or a couple very high fat meals. Avoid raw foods and vegetables that can't be chewed well. And then there's this concept of a small particle diet. So basically in individuals with gastroparesis or small intestinal bacterial overgrowth, or SIBO, which is also very common in Parkinson's disease, oftentimes it's recommended that people avoid fiber altogether because it can cause worsening of GI symptoms. The problem with this is that when we eliminate fiber, we miss out on prebiotics. So prebiotics are like the food for the bugs in our gut. When you take a probiotic, you're taking the actual bug. And when you eat prebiotics, you're eating food that feeds those good bugs in our gut. So by eliminating fiber, you're missing out on prebiotics. And then it oftentimes just leads to a diet that's really high in carbohydrates and is very processed. So instead of changing the content of the diet altogether and getting rid of fiber, we focus on the size, the particle size. And so the concept of this is thinking about foods that are easy to mash with a fork. So pureeing or mashing your vegetables, potatoes, maybe mincing your meats, but just making things literally into smaller particle size, which becomes easier for you to digest. And then try to sit upright or maybe take a brisk walk for the 1 to 2 hours after meals. So that was a lot, but in conclusion, as confusing and as controversial as diet can get, in general, I recommend minimizing processed foods and sugars. Eat a diverse diet because this helps to create a diverse and robust microbiome. Minimize your raw vegetables. Eat organic when you can, especially the Dirty Dozen that can be found on the Environmental Working Group's website. Eat fermented foods or drinks, and then think about this small particle or foods that are easy to mash with a fork. I get a lot of questions on what supplements should be taken. So I thought I'd talk about microbiome and kind of gut targeted supplements. I think digestive enzymes can be really helpful for people, especially, again, if you're someone who, a lot of us just don't take the time to eat very slowly and to chew our food very well. So if you know that that is the reality of your day, digestive enzymes can be really helpful. Horse chestnut is something that you can take just on its own. You can just buy a horse chestnut supplement, or there's a proprietary supplement called Atrantil that includes horse chestnut as well as some other things. And what horse chestnut can do is that it can reduce methane production. Methane is responsible for a lot of gas and bloating and constipation and upper GI pain and distension. And a lot of individuals with small intestinal bacterial overgrowth have increased methane production. So it can decrease methane. It also has saponins, which can help to bind bad chemicals inside our GI tract and help us to eliminate those through our poop. And then it has flavonoids, which are kind of an anti-inflammatory antioxidant. Glutamine is something that is decently common and you'll see recommended in a lot of spaces. The thought is that it can help with gut permeability or leaky gut. It can be protective and helpful to repair the gut lining. I think it's safe and it may help with permeability as hypothesized, but there's just not strong data. So just know that, it's not super evidence backed. And then the last is probiotics. And so let's talk about this. There have been a few studies in Parkinson's patients that have shown multi-strain probiotics can increase the number of bowel movements that someone has per week. Importantly, one of these studies, the company that made that proprietary probiotic was the one who conducted the study. And then there was one study that looked at fermented milk that contained multiple probiotic strains as well as a prebiotic fiber, and showed, again, increase in the number of bowel movements per week. Similar to fermented foods, though, is that a lot of people actually find, paradoxically, that they have an increase in the number of upper GI symptoms. So they have more bloating, more gas, more distension, and more pain from taking probiotics. And then there's this theoretical risk that's talked about in the GI community, of potentially unfavorably altering the microbiome composition in the long run. Again, I don't think this is something that has been shown, but it is hypothesized or a theoretical risk. So in general, what I recommend is that if you're someone who takes a probiotic and you don't have any negative symptoms, and you do notice an increase in bowel movement frequency, that it helps you poop more regularly, then great, you're lucky, and go ahead and keep taking the probiotic. But if you're someone who takes the probiotic and either has more bloating or pain, know that you're not alone, that's not uncommon. And two, if you take a probiotic and you don't notice anything, you don't notice that you're pooping more regularly, then don't worry about stopping and saving money. And I don't want anyone to feel like they're missing out on this like really pivotal part of changing their microbiome, because we don't think that that is the case. Okay, what can we do about constipation? As you guys know, constipation is really the most disabling symptom for most Parkinson's patients. And it plays a huge role in the quality of life for people with Parkinson's disease. Hydration, exercise, of course, are helpful, but oftentimes, people do that and are just still running into a lot of trouble. So that is where keeping fiber and having the small particle diet is really important. And then a lot of people find that a manual massage works really well. So what you want to do is basically kind of outline where your colon is and manually massage your colon, importantly in a clockwise direction. A lot of people like to do this at night when they're lying in bed, and they find that it helps them to have a bowel movement when they wake up the next morning. And then supplements specifically for constipation. So there's many forms of magnesium, and they're all thought to have different roles. But for constipation, you want to specifically look for magnesium citrate or magnesium oxide. The side effects of these can be that it causes watery diarrhea or cramping. So if you take them and you have GI discomfort or cramping, of course, stop or decrease the dose. And then other things that you can take are triphala, which is like a very gentle kind of helps with GI motility and cleansing, so to speak. You can take this every day, regularly, long-term. It's very safe. You can also take aloe in capsules. I think tends to be more effective than the juice that you can get. But aloe capsules can be taken as needed. You don't want to take it regularly because it's more of a stimulant. But for severe constipation, you haven't had a bowel movement in 4 days, aloe capsules can be really effective. Okay. So we made it to talking about the study that we are conducting at Stanford. And so generally, I told you that we know that the composition of the microbiome in people with Parkinson's disease is different. And so we're trying to better understand what role these changes have in the pathogenesis and the development and the progression of Parkinson's disease. So just to kind of review what we already know, we know that there's increased production of microbe-derived inflammatory molecules in the gut of people with PD. We know that in Parkinson's disease, the gut-blood barrier and the blood-brain barrier are compromised. So again, I think most people are familiar with the term leaky gut, but we have the layer between the gut and the systemic circulation or the blood. That's the gut-blood barrier. And then the blood-brain barrier used to be thought of a very privileged barrier between the brain and the systemic circulation. I think we're learning a lot more about the blood-brain barrier, but in general, they're very similar in their anatomy and their makeup. And we know again that in Parkinson's disease, the integrity of these barriers are compromised. We also know that too much inflammation in the brain is, of course, harmful to the brain. It can cause degeneration of neurons, and we see this in Parkinson's disease. So what we're really interested in with that in mind is inflammation and barrier integrity. Our hypothesis is that certain microbes in the gut that are increased in people with Parkinson's disease lead to enhanced barrier permeability, i.e. more leaky barriers, which allow microbial molecules to pass into the blood, come in contact with the blood-brain barrier, alter the blood-brain barrier, and then those microbes and their molecules can enter the brain where they cause neuroinflammation and neurodegeneration. Again, major premises are understanding inflammation as well as barrier integrity, being the gut barrier and the blood-brain barrier. And we're specifically focused on metabolites. So the metabolites that the microbes in the gut are producing. So we are recruiting people with Parkinson's disease. Unfortunately, you're not eligible if you have inflammatory bowel disease or are currently being treated for cancer. And then we're also recruiting people without Parkinson's disease. So in most cases, this is like a significant other or a family member that lives with someone with Parkinson's disease. I told you that, you know, we know that there's many other things that contribute to someone's microbiome, like diet, pet exposure, do you drink well water, what medications you take, etc. And so the thought is that by enrolling people who live in the same household, we're controlling for a lot of the environmental factors. And people without PD are not eligible if they have any of these other neurologic disorders that are listed here. What we're asking for is one blood collection, an at-home stool collection, and then this 20-question survey. And again, so part of what is unique and powerful about our study is we're the largest study that we know of ever to pair both stool and blood. And so the benefit of this, again, is that we're looking at metabolites in the blood. I mentioned the barrier permeability and inflammation. There's a lot more downstream studies that we hope to do from this. We are also collecting from this blood, we're collecting something called peripheral blood mononuclear cells, which will allow us to better understand how the immune system is involved and how gene expression in immune cells is altered in Parkinson's disease. So yeah, we're pretty excited that it's the largest study to collect both the stool and the blood. And we think that that is really unique and helps us to better understand the mechanisms of the microbiome. And then, like Monique mentioned, I wish that we could, give more appreciation, but we're giving a $40 Visa gift card to everyone who participates for their time. And so for your guys' group, we can have people come to Stanford. I got a question about people participating remotely. Unfortunately, we're not set up to do blood draw in places outside of Stanford or with these mobile phlebotomy events that we're organizing. So, people who live outside of those areas can't donate blood, which again is part of kind of the power of our study. That said, if people are remote and they feel very strongly and are really interested in participating, then I hate to say no. And I think it's still valuable at least to have stool. So if that's the case, there will be information on who to contact and we can try to figure something out. Okay, so then, generally speaking, what we're hoping to learn from your donation and your participation and your time, so many things, but as you guys know, there's a lot of, we need ways to better diagnose and detect Parkinson's early. And so biomarkers is a big thing. We wonder if there's a microbial signature that can predict that someone is going to develop Parkinson's disease. Other things are symptom management. Can we give personalized diet advice based off microbial signatures and microbiome? Can we selectively eliminate or add certain bugs or certain metabolites that they make in order to address and improve constipation or GI symptoms? And similarly, can we selectively eliminate certain bugs or metabolites in order to change disease progression? This is just kind of a small list of the many questions that we have and hope to learn from kind of future iterations of pieces of this study. So this is the contact information for my research coordinator. And this, of course, is all going to be an email, and so much other information on how to follow up on this will be in the email. But with that, this is our team. So this is Erin, is a research coordinator who just actually left to go to medical school. This is Mina, who is a PhD candidate and does a lot of the bioinformatics and data analysis. This is Gabriella, who's the research coordinator that you guys will be talking to. And then this is Dr. Ami Bhatt, who is the PI and whose lab I work in. And then this is the Stanford Movement Disorders team.
That's great. Thank you for that great presentation. It looks like you have a wonderful team there.
We do.
Of supporters. That was a lot of information, and there are definitely a lot of questions coming in the chat. I think I'll start off the conversation by that last bullet point that you had on the slide about ways to sort of alter the progression of the disease, things that you might learn from in the microbiome. I know that's probably something that people are wondering about, is because I know there's a lot of talk that that the microbiome, that sort of dysbiosis could lead to the development of Parkinson's. But once you have Parkinson's, can you change your microbiome, and can it change the progression of the disease? So what do you know to date about that? Is there anything that you can share?
Yeah. So in general, the answer is we don't know. But part of the big picture of this is that the microbiome field as a whole needs more mechanistic understanding. And so much, until the last couple of years, so much of the studies, so much of the research had been on just figuring out that there was a difference in the bugs. And so now everyone is shifting again to try to figure out what they're doing and how that impacts us. So I think until we have a better understanding of mechanism, we can't necessarily say. And microbiome, it's an ecosystem. So I think we're not probably going to get many answers by focusing on one bug, because each bug interacts with each other and all the metabolites interact with each other. So there's these complex models of interaction amongst them all. But the original question was progression. I think in starting to understand the interactions of the ecosystem and the mechanisms of the ecosystem, then we can at least find points of areas where we could intervene and change downstream effects, if that makes sense.
Yeah, I think that does make sense. And are there sort of good bugs and bad bugs? And in people with Parkinson's, there's more sort of bad ones, or is it more nuanced?
That's correct. I mean, it is more nuanced than that. Part of there's been, of course, a lot of advances in technology. So even the early microbiome studies were using something called 16S sequencing. And so the technology has advanced a lot. What we use is genome sequencing. So it allows us to look at things more down to strain level, which is a lot more specific. But to answer your question, we had limitations even to knowing completely what bugs were there. So there are putative good bugs and putative bad bugs, but then there's a lot of bugs that we don't really know what they do. And, maybe bug X, when interacting with this bug, is good, but when interacting with that bug is bad. Very little about microbiome is black and white, for sure.
And you mentioned toward the end of your presentation, the concept of a microbiome signature or gut signature.
Yeah.
I'm sure that's a very scientifically rendered value or series of values, but is that something that you're going to be able to sort of publish to say, if somebody comes in, you can take a blood sample or some such and be able to say you've got an excellent.
Yeah. So for that, we've started to just play around with machine learning models to see if we could predict again, based off my microbial signature, who has Parkinson's and who doesn't. I think it's in, for us at least, I don't know who else is doing that. For us, it's in the very early stages, but it's definitely something. I mean, the question of like, could you have a biomarker that's either based on stool sequencing, looking at microbiome sequencing, or metabolites in the blood that is predictive of Parkinson's disease?
Great. And Todd, I'll let you take a question from the Q&A, but I wanted to let everybody know that I am going to go ahead and put the link to the interest form in the chat. So if you are interested, you can just fill that out. And then I think Gabriella will give you a call. Is that right, Dr. Bianca?
Yep, exactly.
Okay.
Yeah.
We do have tons of good questions, a lot of
Yeah.
smart
Yeah.
people in our membership. So
I can
they
just
sort-
scroll through some of these.
you can see them. You can see them yourself. Yeah. And,
Yeah.
and in large part, they sort of start at the top of your presentation and roll down
Yeah.
especially
Yeah.
through it. I didn't want to interrupt your flow, but Jodine's got a couple good and semi-technical questions about alpha-synuclein that you started off at the top of the presentation mentioning. So maybe answering that one.
So the normal function of alpha-synuclein, I actually don't know what the function is. It is not always pathological. And so this is similar to kind of where the field of Alzheimer's disease is running into with tau. But I don't know that it has. I don't know is the short answer of that. But it's only pathologic when it begins to aggregate. And the trouble is, again, and we see this with tau, is that there are people that have aggregates that are not symptomatic. And the question is that like prodrome, or is that not clinically significant? What happens if we remove the vagus nerve, if it affects getting PD? Yeah. So this is always a good question. We don't want to remove the vagus nerve. It is so it's beneficial for us. In so many ways. Yeah. In so many ways. Yeah. So that study, they were not doing vagotomies where they cut the vagus nerve to learn about Parkinson's disease. They were doing it for other purposes, not knowing the implications it had, and then subsequently found out that it decreased the risk of Parkinson's disease. But while it does decrease someone's risk for getting Parkinson's, we don't want to go without our vagus nerve.
And there's also a question following up on that doesn't show causality between the microbiome and the PD.
Correct. No, that's absolutely correct. The conclusion that's been derived from that is that, again, the vagus nerve is implicated and probably critical for the alpha-synuclein that is in the gut to migrate into the central nervous system or the brain.
Mm-hmm.
Is nervous system management, switching from the sympathetic to parasympathetic and back, related to the gut and microbiome? I don't know that I understand that. Gabriel, I don't know if I understand what you're asking, but like, does the gut microbiome alter our ability to switch back and forth, maybe?
Maybe Gabe he could possibly rephrase it as we move on to a couple others.
Yeah. Are fermented foods associated with stomach cancer? So this is interesting. Monique was asking me this, or telling me about this association. I've actually not heard of this. I will talk to my GI colleagues and look into the literature, but it's not something that I'm aware of. If that's the case, I imagine it's much higher doses of fermented foods. And studies have shown now that you can have the benefit in terms of anti-inflammatory effect with like 1 to 3 servings of fermented foods a day. So I would just say, if that's something that, there's even a sniff of in the literature, or you're worried about, just stick with like 1 to 3 servings of fermented foods.
Per week.
Per day. Yeah. The role on intermittent fasting on microbiome health. Yeah, so these are all great questions that I think there just aren't a lot of definitive evidence on. You could probably search PubMed and find some stuff of intermittent fasting on microbiome health. Yes, I think it does affect the microbiome. Whether it creates lasting changes in the microbiome, I don't know. You know, the microbiome can shift even after certain meals. So I'm sure it has some effect, but whether those are lasting effects, I don't know. If you already have PD, should I be worried about buying organic? Again, so much of this, there's not studies that have shown like PD patients eating organic versus PD patients not eating organic, but my recommendation would still be yes. You're still adding, you know, cumulative pro-inflammatory things. And even outside of Parkinson's, you know, pesticides are known carcinogens, just not good for our health anyways. Metamucil for constipation, absolutely. Again, Metamucil is something that I would say listen to your body, because some people respond well to it and some people don't. It's a fiber, so it can cause kind of increase in symptoms, but if it doesn't bother you and it helps you poop regularly, then great. I am not familiar with PS 128. These are good questions. Neurally is a probiotic that specifically targets PD. I don't know, but I'm gonna look into it. I've not heard of that. I can share a copy of the slides for sure. What about DBS? I don't know what Carol's asking about it.
Just to follow up on that last question, to let folks know about the slides, we will try to get those posted on our website, pdactive.org, under past events and presentations section there that we'll send out a link to so people can find it easily.
Thank
There's
you.
a couple people that have their hand up. Should we take a couple questions real quick from the audience?
Yeah.
Sure.
I think Sheila, you have your hand up. If you'd like to ask your question directly, you can. Just unmute yourself and ask.
Come in, Sheila. You're on mute.
We also have a question from, I'll go ahead and unmute Sarah as well. Sarah Armstrong, if you wanted to ask a question directly to Bianca, she's willing to do that. Maybe your hand up was just an error also. And then we have another question from Shoshana Silberman. If you want to ask your question directly, you can do that.
Now lots of hands are being raised. I wonder if there's confusion around the feature. Well, let's keep going down.
Okay, I'll keep going through. If I participate in the study, do you share my individual results? Unfortunately not, because everything is de-identified as soon as we, you know, from the time that we give you the kit for the stool collection, you're already some random number. And so everything is de-identified from the get-go, so you don't get any personalized results. We will, of course, share results as soon as we analyze data and start to publish things, but unfortunately no personalized results to protect.
Any idea what that timeline might be? I mean, how much longer do you expect the trial to run?
Yeah, it's a good question. So we're hoping to end enrollment at least for this phase of the study by the end of the year. Right now we have like 170 participants, and we're hoping to reach 200 pretty shortly by the end of the calendar year. I have a couple of grant applications in, and I'm hoping to get more funding to do, you know, the sequencing studies and, you know, more data analysis. But nothing is super quick. Things take time for sure. I'd say it all just depends, but in terms of publication, like getting through a journal, et cetera, honestly, probably a year or two, but we could share, you know, at least our findings and results with participants. What we've talked about doing and hope to do is just create like informal results to share with participants long before it gets through journals, because journals take forever and all the iterations and stuff like that.
Mm-hmm.
Okay, I'm going to try again with the live questions. So yeah. Larry, it looks like you have your hand raised. Do you have a question that you'd like to ask Bianca directly? Hello, Larry. Can you hear me? If you can hear me and you want to ask your question, it looks like you have your hand raised. Okay. I think Thomas also has his hand raised. So let's try Thomas. Thomas, you're live on the air if you want to ask your question directly to Dr. Bianca.
I wonder if something's just not working for whatever reason.
Possible.
Yeah.
I think the more
I
we
don't think
click
we
on
can talk on our end. There's no microphone here on my end, says Kathleen.
Okay, I'm going to try one more person. Joanne, Ruby, are you there? Would you like to ask your question?
Okay.
She did put a question into Q&A. We might as well field that one now that we've got.
I think
her
they just have
on.
to unmute. Oh, she's unmuted.
Can you hear me?
Yes.
Yes. Hi, Joanne. We're ready for you.
I can't see you anymore because I'm filling out your questionnaire, but yeah, I mean, probably other people have heard me talk about this, but the question has to do with UTIs and E. coli and the gut, and to what degree somebody with recurrent UTIs over the years may have been impacted either causally or correlationally, or what role UTIs
Yeah.
play in all this.
So I would think of it as different from the gut microbiome. You know, as I'm specifically focusing on the gut microbiome, but as I mentioned, the microbiome really, there's a brain microbiome, there's our skin biome, there's our vaginal biome, there's everything. And so I would think of that recurrent UTIs could be many things. One is anatomic. There are certain, anatomic variants that increase one's risk for developing UTIs. But I would think of it more as a change in the microbiome of the urogenital system as opposed to the gut microbiome. I don't know anything about the urogenital microbiome and Parkinson's, unfortunately.
Do you know if it's being looked at very much or at all?
I'm not aware of anyone looking at it.
Exactly.
Yeah, I'm not aware. You would have to, for the reasons I mentioned, there are ways that we think of the gut communicating with the brain. I think that there's less, at least at this point in time, understood communication between the urogenital system and the brain. So that would take an extra layer of kind of communication.
Yeah, good. Thank you.
Of course.
Affirms a lot of things. Thank you.
Yeah, of course.
Good question, Joanne. Monique, I see that Larry unmuted himself. Are you ready for your question, Larry?
Yes, if you can hear me.
Yes.
Dan, fire away.
Is it possible to have the blood drawn at the Stanford in Emeryville or one of the other satellites?
Oh, good question. I don't think so, because what we're doing at Stanford, there's something called the CTRU, which is the Clinical and Translational Research Unit. So all clinical trials at Stanford have to get blood draw through there. It's not through the clinical labs like LabQuest LabCorp or Quest. So I don't think so, but still, please ask Gabriella that, because she is really the brains behind all of the logistics and execution.
It would seem like the other branches of the hospital could take the blood and then send it down there to this organization that you're talking about.
Yeah, I know. So the problem is no one helps us with personnel or funding for all of that. So all of our own funding is what we have from the lab and from grants. So we have to hire someone, spend their, the hospital is not involved.
Gotcha. Thank you.
We'll try to accommodate everybody that wants to participate on Saturday, September 9th. But if for some reason that doesn't work, then we'll work with Bianca and Gabrielle to maybe set up a second event or facilitate another way for people who to participate to be able to participate.
Yeah, we do not want to turn people down. We're really appreciative of people wanting to donate their time and their specimens. So if we can accommodate everyone on September 9th, which we do have kind of a cutoff of how many people we can accommodate, just because our team does all of the processing for the blood sample. Speaking of, money and limited resources, we do all the processing ourselves, and we just can't process like more than 25 or so samples at once. So anyways, long story short is that if there's more people who want to participate than what we can accommodate on September 9th, we'll organize a second event. And that's like a best case scenario. That makes me happy. That's great. So.
Great. Thank you. Thanks for the question, Larry. Andrea, I unmuted you. So if you do have a question, feel free to ask directly.
Yes. What's your position on eating dairy foods? There's some sentiment that it's not good to eat them.
Yeah, so this is where we can just get so lost in the weeds with diet. In general, if eating dairy, I recommend fermented dairy is better. And it depends on what we're speaking of, but like if you're it's tough. If you're someone who can't tolerate, so I would say, taking a step back, most people have some sort of lactose intolerance at higher doses. Just as we get older, we produce less of the enzymes that we need to digest. And then, maybe because of microbiome changes, et cetera, people can and cannot tolerate dairy differently. You've probably seen like a lot of the A2 dairy that's become popular in stores. Generally speaking, that's thought to be better tolerated. So if you're speaking just about tolerance and things, I recommend A2 dairy. There are some pretty good studies if you're looking at, this is controversial, but raw, unpasteurized A2 dairy, let's say from a farmer's market or something like that, if you have a reliable farmer, that has been shown to be beneficial and kind of anti-inflammatory. Some people anecdotally have found like remarkable improvement in their autoimmune disease and things like that. This is again such a, like, you can get lost in the weeds, but a lot of our dietary practices, of course, in the States with the depletion of our soils and with a lot of pesticide use and with the way that we've bred our cows to produce like large amounts of dairy with A1 proteins, it's just harder to get the nutrients that we intend to get out of the foods that we're eating that are meant to give us those specific nutrients. So yes, you can find a lot of literature about dairy being bad for any number of things. Personally speaking, not from a medical advice, I try to just limit dairy. And when I do eat dairy, I try to eat high quality, similar to my philosophy on red meat, that like limit it. But when I eat it, it has a lot of important nutrients. And so when I eat very high quality, which is grass fed and grass finished. Similar with dairy, I drink raw unpasteurized milk from the local farmer's market. That's A2 protein. I don't know if that's helpful, but
well, that's
muddy. It's muddy water.
What I was more thinking of is that there have been apparently some studies that associate increased dairy intake with increased risk of getting Parkinson's.
In our practice, we do not believe that to be truly causative.
I'm so glad.
Yeah.
Okay, thank you.
Really, the causative environmental factors that we feel pretty sure about are some well water, of course, Agent Orange exposure for any veterans, pesticide exposure. Those are like the main environmental factors. We know that traumatic brain injury can increase risk, but we do not think of dairy as being causative.
Okay,
I have
Jodine,
oh, sorry, Todd.
it's okay. I just saw Jodine's hand still up. So Jodine, you should be off mute if you want to ask your question.
Oh, hi. Hey, I just want to congratulate, and I don't know how to turn my camera on either. All my controls look weird. I just want to congratulate you on this study, because it seems that this idea, you know, sometimes these ideas get out there and then it takes a long time to get into like conventional medicine. And this is like really fast. I mean, I, you know, just became aware of some of this stuff. And so I really want to be in your study. And I get information about PD from all over. And I think this was a NOVA, but I'm not sure. They gave mice fecal transplants from Parkinson's patients, and the mice got Parkinson's, quote unquote.
Yeah.
And then they transplanted them with normal people, fecal transplant, and they cured it.
Yeah. Yeah. So I get a lot of questions about fecal transplant. I think right now there was a bunch of stuff that went on this summer, I think it was, with the FDA and fecal transplants.
We can describe what it means.
What
is a fecal transplant?
So a fecal transplant
Am I muted?
is
Oh,
basically people taking a pill. So first they take antibiotics and kind of wash out their gut microbiome. And then they take a pill that's made from usually like dehydrated feces from a healthy donor. And it's supposed to repopulate their microbiome with this new microbiome. It is proven to be like curative for C. diff infections. And that was kind of how it gained traction initially. Right now, the major kind of roadblock is FDA regulation. I think that it's promising. Like, I think that the main question that it's getting at is how do we truly shift one's microbiome? If we know that there's dysbiosis and it's linked to all sorts of diseases, again, what do we do about it and how do we change it? And I think generally speaking, you know, we all started taking probiotics and recommending probiotics, a decade ago or something like that. And now I think the general consensus is that unfortunately taking a probiotic alone is not going to completely change your microbiome permanently. And so the question becomes like, again, how do you change one's microbiome? So that's where this notion of the fecal transplant comes in. I think that fecal transplants will happen and help, but they'll be kind of repackaged into something different and they need to pass regulation. So I think there's promise in it. Right now it's regulatory roadblock. And of course, like safety is a huge concern and the quality of where things are coming from. There's like a whole black market for fecal transplants that exists, where, I mean, you can go online and find communities of people that are doing things unregulated and without any medical professionals or anything. But yeah, I think that it's promising. It's just so many things have to be worked out.
I'm really curious about who those normal donors are in these protocols.
Yeah.
Like you're saying, you know, I guess the FDA needs to look into that. But I think now that it's approved for C. diff, people are selling it, you know, off label.
Yeah.
If you're like real keen to have it,
Yeah.
but I have no idea you know, who the normal donors are. One really quick thing.
Yeah.
Are mononuclear cells the same as monocytes?
Yes.
Oh, okay. Because I've had this is a personal thing, sorry, everybody. I've had elevated monocytes for a very long time in my blood studies. Is that something that you look at in the study, or is that a qualification or something?
No. It doesn't disqualify you. No.
Okay.
No, it doesn't.
Thanks.
Yeah. Someone asked, how can we increase short chain fatty acids?
Question.
Yeah, it is a good question. So, generally speaking, eating prebiotics and fermented foods should increase one's short chain fatty acids. Eating prebiotic foods, and if you just Google which foods are high in prebiotics, it'll come up with a list of what you should eat. You can take a prebiotic supplement, and fiber is really important for increasing short chain fatty acids. Now, of course, there are certain bugs that produce short chain fatty acids. So theoretically, if your gut is depleted in the bugs that create short chain fatty acids, you know, you just are going to reach a limit in terms of what you can produce. I think I should have looked this up. I think that there are short chain fatty acid supplements you can take. There are, because butyrate is one of them. Yeah, so you can take short chain fatty acid supplements. Butyrate capsules are actually oftentimes used. I have seen trickles of some literature, then it's questioning whether like repetitive supplementation of butyrate could actually be detrimental. So my conclusion with a lot of this is, again, like more of a good thing is not always a good thing, as it relates to fermented foods, probiotics, prebiotics, whatever. I would have to look up kind of what data there is in Parkinson's patients taking butyrate or other short chain fatty acids. But there's a couple answers for you.
Great. Boy, the questions just keep flying in. There's still some hands raised. We'll come back to those after we field a couple of typed in questions. Joe asks about companies that are out there already that analyze your microbiome.
Yeah.
Do you know of any
Yeah.
of those?
So there's a lot. And again, the question with this becomes, truth be told, this is where like we don't necessarily know what to do with that information or how to change it. So I feel like right now so a lot of functional medicine doctors are doing this. People are paying $800, $500, whatever it is, out of pocket to have their microbiome tested. And it might show, you have an increase in this bad guy and a decrease in this good guy, but what do you do about it? We don't have targeted ways of decreasing certain bugs. And so to my knowledge, knowing that is not going to change the recommendation. Like my recommendations are still going to be to eat fermented foods, to eat prebiotics, to include fiber, to minimize processed foods, to minimize sugar. So if you're a data person, which is fine, and a lot of us are, and it might just help you and it's interesting, it's kind of more of an academic exercise than anything that's going to change recommendations or give actionable results at this point in time. Hopefully that's different, in a couple of years, but.
Okay. Let's see. Let's try a person with their hand raised. I see Carol Scott. Carol, I'm going to click the button that says allow to talk. So if you unmute yourself, you can lob your question out.
Yes, I'd like to know if you can join your study if you've had DBS surgery.
Absolutely, yes.
Thanks for asking that, Carol. I see
Yeah,
that.
thank you
Okay,
for
that's what
asking
I want to know.
Yeah, thank you. That's a really important
Thank
question.
you.
Yeah.
Good. Thanks, Carol.
Speaking of this study, did anybody who filled out the interest form have any problems or questions while doing it? Now would be a great time to let us know if the form was not working or. if you
And
have
that
any
interest form
questions.
again is a link that Monique put into the chat window.
Yeah, and I'll put it in again right now.
Yep.
And we will send it around as well. Okay, sorry about that. Todd, go ahead.
Just a tactical question here. Dana's asking about carpooling on the morning of. That's great. We will try to facilitate that, but it is dependent upon the scheduling through the interest form. In other words, you will probably be signing up for 15-minute slots. And so if you can coordinate with others in the PD Active membership group to sort of select the adjacent 15-minute slots, that would facilitate carpooling. So we'll have to leave that up to you folks, but I like the question.
Other tactical questions. So people who don't have a significant other that lives with them can absolutely still participate. And that's on both ends. So if you're someone who doesn't have Parkinson's disease and you live with someone who has Parkinson's, but that Parkinson's person doesn't want to participate, you can still participate, and vice versa. So we're accepting people who are not paired on either end of that. So Parkinson's patients without a healthy partner and healthy partners who don't have a Parkinson's partner that wants to participate.
Oh, okay. So I can participate even if my person with
Yeah.
Parkinson's doesn't.
Yep, yep.
Got it. Great.
The most ideal is that we get pairs, but either way works for us. Yeah.
That's
Cool.
great.
That's good to know. So on a similar vein, the North Berkeley Senior Center is off of University and has public transportation to get there, easily accessed that way. So use that Google map to get public transportation to the facility.
I think it's located about MLK and Hearst, Milvia and
Yes.
Hearst, and around that area.
A block
And
or two.
we'll send out all the details shortly as soon as we get that confirmed.
Lots of good kudos here, Bianca. Thank you. Enlightening and helpful. Important. Does beer count as fermented food?
I was just seeing that. I get that question a lot. It does. It
Yes.
does. Yeah, it does. Absolutely. It is fermented. And yeah, you don't want to heat your fermented foods. The other just quick thing I'll say about like sauerkraut and things, you just have to, this is a lesson we've learned through everything we eat today, is like you have to read ingredients for the most part. So a true fermented sauerkraut is going to be refrigerated for the most part. There are some that are not, but it does not contain vinegar. So the sauerkrauts that you get on the shelves that are not refrigerated and are like cabbage in a bunch of vinegar, it may be minimally fermented, but it's not the true really good fermented. You want to get the ones that's just like a pickle or sour or cabbage in a water brine that has become fermented. So no vinegar is better.
Good to know. That's
And
important.
there's I will go ahead and just put a plug in for my favorite pickled product, people. In the Bay Area, it's the Golden State Pickles Company, and they sell at the farmers markets on Temescal on Sunday. They sell at this little shop called Preserved in
Nice.
Temescal, and I think maybe sometimes at the Berkeley Bowl. But she makes really, really wonderful products. So I just put that in the chat as well.
Bianca, a question from Nancy. Is the mitochondria affected in any way?
In Parkinson's disease, yes, as in like most diseases, but I don't know like the relationship between the microbiome and the mitochondria.
Okay.
Is it possible to talk, and we only have a few more minutes, but I'm wondering if you could talk just for a minute more about the relationship between the inflammation and the microbiome and Parkinson's. You know, is the inflammation, the relationship with Parkinson's all sort of mediated through the microbiome, or is there other inflammation that's involved?
Yeah, I think it's both. So I think that there are a variety of reasons why one can have inflammation, and there's various places that can be inflamed, right? So there's enteric inflammation, which is inflammation inside the gut. There's systemic inflammation, like we measure blood markers, ESR, CRP, can measure various cytokines or interleukins. And then there's inflammation in the brain that right now we don't have any way of measuring except for postmortem studies when someone passes and they do a brain autopsy. So there's various pockets where inflammation can be. We know that neuroinflammation, a little bit of inflammation is good in acute issues when you need immune cells to go to some like sprained ankle and, whatever, but generally speaking, chronic inflammation is not good. So goes without saying that chronic inflammation in the brain is not good, can cause neurodegeneration. There are probably many reasons why people have increased inflammation in the brain. But as someone who's studying the microbiome, what we're trying to understand is how might the microbiome and these changes that we see in Parkinson's disease microbiome contribute to inflammation in the brain. And so again, what we think is that the increased inflammation inside the gut because of this shift in the microbiome in Parkinson's disease causes leakage of the gut barrier. And then all of these inflammatory molecules go into the blood and they come in contact with the blood-brain barrier. And since there's so much similarity in the blood-brain barrier and the gut barrier, we think that what causes leakiness of the gut barrier causes leakiness of the blood-brain barrier. And then you're getting things inside the brain that are not supposed to be there. And I neglected to mention that we have an in vitro model of the blood-brain barrier. So we're actually specifically testing metabolites that are made by the microbiome and found in the blood and putting them in a petri dish and testing them on the blood-brain barrier and seeing which ones cause leakiness of the blood-brain barrier. So that's like the, what we're doing specifically with this study. And that's why the blood is so important, not only to look at the immune cells, but to look at those metabolites and then testing each individual metabolite on the integrity of the blood-brain barrier model that we have.
Oh, interesting. That's very helpful. So it's those molecules that are kind of getting into the brain that could of the alpha-synuclein.
Yeah. And even if they're not directly causing aggregation of the alpha-synuclein, if they're contributing to neuroinflammation, then they're causing toxicity to the neurons and neurodegeneration.
Interesting. I thought it was interesting that you said that serotonin was made by the gut bacteria. I had no idea that.
Yeah.
Dopamine's not involved in the gut directly, right?
There may be a little bit of dopamine that's made in the gut, but the majority of it is not. I think the majority of serotonin is made in the gut.
Very interesting.
Yeah, some neurotransmitters are made in the gut. It is so interesting. Yeah.
That's really interesting.
Yeah.
I wish we could make more dopamine in the gut and anywhere else for that
Right?
matter.
Yeah.
Feeling a little low right now.
People call the gut the big brain, actually, because there's more neurons in the enteric nervous system than the brain, I believe. But it is often a neglected or kind of forgotten organ and part of our nervous system.
The gut, yeah.
Yeah.
So Bianca, we're at the end of the window here. Are you okay to just go for 5 more minutes for 1 or 2 questions before we sign off?
Yeah.
That's nice. Rosie, I gave you the ability to talk. If you still have your question, fire away now. You're unmuted.
Okay. You might have mentioned this, but I did hear that the slides would be available, but will the whole webinar be available as a recording?
Yes, we will send out a link to the webinar, to the recording, and to the presentation, and to some extra links that have been mentioned in the talk tonight, as well as the interest form that we've posted so that you can choose to participate in the trial if you would like.
Okay, thank you.
You're welcome.
I'm pasting that YouTube link in the chat right now for everyone.
Oh, okay. That's the YouTube link where we post all of our webinar recordings.
Yeah. Thanks, Rosie.
Let me answer Michelle's question quickly. So people with irritable bowel syndrome, IBS, are not excluded. People with inflammatory bowel disease or IBD are excluded. And that's because that's its own inflammatory autoimmune disease within the GI system. And we don't know at this point how to kind of interpret any changes that we might see in that microbiome with what we're trying to understand. But that's, a design of our study. There are other PD microbiome studies that have included people with IBD. That's just something that we decided. But to be clear, it's IBD, that's exclusionary criteria, not IBS.
I hadn't realized there was a difference.
Yeah. Inflammatory bowel disease, IBD, is an autoimmune disease. Usually people are on biologics, disease modifying agents. Inflammatory bowel syndrome, IBS, irritable bowel syndrome, I'm getting them all mixed up. Irritable bowel syndrome, IBS, is kind of a nondescript way to describe people who have GI symptoms, GI discomfort, constipation, diarrhea, pain, bloating, gas, etc.
How would I know which one I had?
It's a good question. If you haven't been diagnosed with inflammatory bowel disease, it's probably IBS. I mean, IBD is generally diagnosed by a colonoscopy. But I think if you've talked to a doctor about your GI symptoms and no one has raised concern for IBD and no one has treated you for IBD, then you probably have IBS.
Okay.
Interesting. Is that good for you, Michelle?
Yeah, I probably have IBD,
All right. Great.
unfortunately.
I'm
Oh,
sorry to hear that.
I'm sorry. Yeah.
Couple more quick ones here I'm seeing in the Q&A feature, Bianca. Kaipo asks, would you recommend the use of turmeric to reduce inflammation? And then Karen has a quick one, which is how does alcohol affect the gut?
Yeah. So a lot of these, it just depends, you know, how much evidence you want before you decide that you're going to use something. So turmeric and curcumin are anti-inflammatory. You know, they're very common in a lot of Eastern medicine. My kind of fallback on a lot of this is like, there's unfortunately just not great data and studies on a lot of these supplements because pharmaceutical companies honestly fund most research and they don't fund supplement studies. So there might not be robust evidence for it, but I think as long as you, one, can afford it. Like I always get a little leery when people are spending hundreds of dollars per month on like, you know, 10 different supplements, thinking that it's going to make a huge difference. I think less is more. And it's really also about the quality of your supplements. So, you know, Amazon sells some good brands, but a lot of the stuff on Amazon is just totally not regulated. They've shown before that like there's a variety of different doses that's in any one pill. Sometimes there's unknown ingredients in the pills and stuff like that. So it's about, to me, it's about quality of the supplements. And it's just, are you comfortable spending your money on something and taking something that may or may not really be doing something? Yeah. Alcohol affect the microbiome. I don't think that I know like specifically which strains are impacted. I have not seen anything where there's beneficial effects. I mean, beer, as we said, is fermented, so there's that. I couldn't speak to wine and liquor's effect on the microbiome. I don't know specifically.
There was another question about the ketone diet.
Yeah, great question. Again, I don't know that anyone has studied ketone diet effect on the microbiome, kind of similar to intermittent fasting. I mean, I imagine that it certainly alters the microbiome, but in what way specifically, I don't know.
Okay,
Yeah.
thanks.
Yeah.
All right, I think it's time to wrap up. 7 minutes after. Thank you for staying over with us. And thank you for joining us. It was a very informative presentation, evidence-based, as one of the members said. So
Yeah, I'm sorry there's not, you know, it's a lot of good specific questions. There haven't been studies on a lot of these things. I mean, the microbiome field is somewhat at its infancy, not totally at its infancy, but it's definitely novice. So, you know, there's not a ton of people that are studying specifically how do ketones affect the microbiome or how does intermittent fasting affect it. We suspect that it has some, but there's just a paucity of evidence for a lot of these specific things. But great questions overall. Sorry I couldn't answer them.
we're happy to have you here. You're a revolutionary.
Thank you for saying that.
Thanks,
you.
Bianca. And one other quick point is to the whole pediatric community, they only need 30 more to hit their 200 mark. So let's see what we can do to help them get to that milestone.
Thank you so much.
Thanks, Bianca. Really terrific.
And please reach out to me if anybody has any questions. And also Gabriella Green, Bianca's assistant, also can answer any questions. And we'll put her name in the chat one more time so everybody has it. She's available to answer all of your questions. Let me put that in the chat one more last time.
Doing that now.
And then we will sign off.
Thank you.
All right. Thank you so much.
Thank you guys. Have a great night.
Okay. Bye-bye.
Thanks.
Bye.
Bye, Bianca.
Bye.